Hi, good morning. Uh, we'll start in around 10 minutes. Thank you for coming.
Morning.
Hi, good
Hi, good.
morning. How are you? Thank you
I'm
for
fine. Of
coming.
course. So, were you able to add him as a co-host?
No, um, we need to. Hi, Johannes. How are you? Thank you for coming. Johannes will be a guest speaker in the future. So, welcome.
Welcome.
If you want to speak in a room, like right now you're in the audience, you would need to press the hand raising button if you want to chat really quick with us. It's next to the microphone symbol. There's a hand raising option, or I'll invite you to speak. Oh, yeah, there you are. Hi, Johannes. How are you? The unmute button is all the way on the bottom right. Um, there's a little microphone symbol.
Oh,
Yeah,
And
you
yes.
got it.
Sorry, I
yes,
think now I got it. Now I got
Yes, you
it.
got it.
Thank you very much. I'm very happy to meet you.
hmm,
Sorry for the technical problems.
oh, no worries. Nice meeting you too. How are you today?
Very fine, very fine. Thank you. And you?
Good, good. I'm actually
I'm
on vacation,
okay. That's fine.
but
That's good.
that's
But
good.
science keeps on, also in vacations, right?
Exactly. Doesn't Science never stops, right?
Perfect.
No.
Hi, Jamie.
Yeah. So,
Hi,
Johannes,
hi everybody.
you
Sorry.
found the most important options here, and we followed each other. I will send you later a link where you're a speaker because now that we follow each other, I
Thank you
can
very
add you
much.
as a speaker. So
Perfect.
yeah,
Thank you.
Johannes will be coming later on, and he will talk about how neurons control feeding behavior at cortical synapses. So it will be really interesting and actually kind of related. So, are you interested in listening to the talk today?
Yes, I would like to listen if it's possible. Yeah.
Wonderful. Yeah, of course. We are always happy if people would like to listen, so that's perfect. Yeah, meet Rahim, meet Jamie. They are both here also moderating the rooms with me. So
Very big pleasure to meet you. Your paper is fascinating. I'm
oh,
Jamie.
Johannes is not the guest speaker today. He will be a guest speaker
Oh,
um, later on. But
ah,
he just made an account. We just followed each other. Um, it will be on July 20th, the talk.
excellent.
Yeah, and this is good
I really look
practice
forward to it.
now to, kind of figure out the platform as well.
Hi, Katie. Hi, Frank. Hi, Suhil. Hi, Sarah. Welcome. We will start in around 7 minutes. We started the room earlier, so our guest speaker can, figure out how to join and to share the link. So yeah, thank you everyone for being here, hanging out.
Hey, did you see my messages?
Well, let me check. Cecilia, do you know if it's still the case that that you have to first log into your phone and only then the desktop app works?
Oh, no. All you need is your phone to give you like the sign in code. You know that code that he asks for. It says put in your phone number and we'll send you the code. So that's it. So your phone will just send you that code and then you can enter it on the desktop app. So you don't necessarily have to sign in on your phone.
Okay, because he says he's having trouble logging in. So yeah, I said to maybe just try it first on the phone and then try the desktop app again. So, but we still have a couple of minutes. So thank you.
Yeah.
Hi, Gilbert. How are you?
Hello, how's everybody doing?
Wonderful. How's
Yeah.
your summer going?
Hi Dee, not bad. Sorry, hi. How are you?
Good, good, I'm doing good, actually. I'm happy to be here, finally, on time.
Yeah, in the meantime, you can meet Dr. Johannes Vogt. He will be a guest speaker on July 20th and came to join us today to listen to this talk, which is very cool.
Doctor, it's a pleasure to meet you. I'm looking forward to your talk.
Doctor, you gotta unmute if you're speaking. You gotta click the mic. Oh, maybe he's not close to his phone. Katerina, if nothing else, I guess Dr. Maldonado could just sign in on his phone.
Yes, exactly. That's what I suggested. So, let's see.
Hey everyone, I know it's 9 o'clock, 9:01, but just bear with us. Our guest speaker's still having some technical difficulties. Hopefully he'll be here soon. Hello, welcome everyone. Thank you for coming. We're going to start in a moment. Our guest speaker is still trying to figure out the app. The desktop app, for some reason, is not figuring out something. But we're going to start soon. So thank you for coming. In the meantime, you could go ahead and read the paper that's posted at the top. That's the paper that we'll be discussing today.
I would encourage everybody to have a little look because it actually is quite fascinating stuff. I'm looking forward to hearing this doctor's talk as soon as they're up and running. And thank you everyone for being with us.
He's saying that he's not allowed to enter. I'm not sure why that would be. I um, never heard of it. Um,
Does he follow you?
no, he's just apparently making the account now. So,
Oh no.
but
When
he's
it's
allowed to enter the room, right?
when he said it doesn't, you're not allowed to enter. Was that like access denied as he made his account?
I
Is
don't
that
know. He
allowed him to
just
make?
wrote
Okay.
he's not allowed to enter. Um,
Oh.
Well, do you have his phone number? Maybe you could speak to him on the phone or something. I don't know.
yeah, let me. Yeah, if I drop out, I'm on the phone real quick.
Yeah, of course, of course.
So this talk is Dr. Maldonado's food addiction vulnerability in microRNA signatures. So this is for everybody who's interested in microRNA. I know I am. I don't know nearly enough about it as I'd like to, but this is what these things are for. Um, are you interested in this, Cece Rahim, with microRNAs?
Yeah, I am. But actually, the part of what I'm more interested in it, I think you'll be fascinated to learn is Dr. Maldonado actually does research on certain things like depression, anxiety, eating disorders, and things like that. And that's kind of the vein that I'm hoping to get from this. So, yeah, the perspective, and I think it's going to be really interesting looking at it from that point of view.
Most definitely. Isn't it really interesting when we actually find the biological mechanisms that's making things difficult for people? I think there's so many people that can take comfort in knowing that there could be something out there to help them along with this rather than just feeling adrift in it. It's a very big problem. I even said in the paper that it has a big problem.
Yeah, exactly. And sometimes having some solid research behind something like this, it does help with maybe naysayers or people who are like, oh, this doesn't exist, or this is, I don't believe in a condition like this. So having research like this definitely does add some credibility to people who might be going through something. So yeah, I like that.
Not just credibility, but hope, right? Because if it's something that gets acknowledged and it's something that's getting the attention to give people a helping hand there, a lot of times you can psychologically get through a lot more knowing that there's a reason behind it or at least some cause as opposed to just be told, oh, you have this, and we don't know what it caused. Anyway, have a nice day.
Yeah, exactly.
We've had a few rooms on microRNA as well in the past, which anyone listening to this should see about checking them out. And they've always been really fascinating to me, as these like things that we haven't been given enough attention to, and yet might actually have a big say in what happens with our DNA and everything. That I've always found that to be incredibly interesting.
I agree. I definitely agree with that.
Do you deal with it much in epidemiology, Cicerine?
Oh, sorry, I was away. Um, yeah, so the beauty of epidemiology is, we get to actually investigate the disease and kind of provide um, almost a trail of where the disease came from, what are some of the things that affect the um, spread, what are some of the things that make it worse or better. So, yes, we do deal with some of that there, but yeah, with just people not understanding or accepting the evidence. I think that's, even during this pandemic, you see people not accepting the evidence of the existence of things. So, yeah, but thankfully, we do have evidence to show. So, I'm happy about that.
I'll agree to that. Yeah, I think we can all agree there. For everyone that's been popping in just now, welcome to Science Society. We're just in the process of helping our speaker set up with Clubhouse. So if you can just bear with us, we will be soon talking to Dr. Maldonado, and the topic is food addiction vulnerability in microRNA signatures. Um, so we're mostly just relaxing and just chatting for a little bit until everything's all fixed. And we are very grateful to have you here and very grateful for your patience. Thank you.
Yeah, and for those of you who, you know us, this is very unusual for us. But this is technology and we do have technical difficulties every now and then. So I guess it was bound to happen, but this is that one time that it happens. So yeah, just, but this is, the Science Society and we're always on top of things. So just wanted to but thanks for coming though still.
Absolutely. I think every single person in Clubhouse who's here at the moment will all be nodding in agreement with regards to some technical issues from time to time with this app. As Cece Rahim was saying before, the paper is pinned up in the room. So if anybody's curious to have a little read of that, just to get a little bit of background information, they can do. It's quite interesting. There's even a little video attached when some of the people involved were talking about what they were doing. So anybody who wants to have a little look at that can do. Hi, Dr. Shah. Nice seeing you here again.
Hi Jamie, hi everyone.
So as you can see, Katerina is on the phone with Dr. Maldonado, so they're talking things out. Hopefully everything gets sorted out. I don't know what's happening with Clubhouse today. Dr. Maldonado literally cannot find the room. It was not allowed to come in. And so, yeah, but hopefully that gets sorted.
I'm going to go on a slight tangent for a second and talk about these little pictures up here, how you've got the microscope for science and the red apple for health. A white heart for addiction? Is that appearing the same to everybody else?
Yeah, it's the same. I don't know why the
It's
apple,
like
I guess an apple a day keeps the doctor away.
that's what I thought the apple was. That's exactly what I thought. But the pizza with engineering, I have to really squint to try and imagine that. Maybe because I don't know, pizza's an incredible example of human engineering. I don't know. Um, but white heart being addiction, I don't even. Does a white heart symbolize anything that anyone can?
I'll look it
Um,
up.
yeah, that I'm not understanding. If anyone in the room has any ideas of why there'd be a white heart for addiction, just drop it in the room chat and we'll we'll hear what you think.
Okay, so some sort of solution has been arrived at by Katerina, so hopefully this works. Let's see what happens when she comes back. But speaking of the white heart, so it says the white heart is used to represent love, support, close bonds, and admiration for things that have some relation to the color white, such as white colored clothing or animals. I don't know. It just means pure love. I guess it's just a supportive thing to, for someone who, is struggling from addiction, maybe.
Oh, interesting. So the symbol is a more of a symbol of the positive response to addiction as opposed to an image that portrays the thing, like microscope meaning science.
Yeah, because
That's
here
interesting.
it says, what is the color for addiction recovery? And it's purple. The official color for National Recovery Month is purple. So not white.
Oh, there you go. See, this is why I come to Science Society, because every single time I'm here, I always learn something new, and I hope that's the same for everybody here.
Yes, no, definitely. Um, and one more time, this is one of those rare occasions where we have issues like technical difficulties. Our speaker, Dr. Maldonado, just could not get into the room. We don't know what's happening with Clubhouse, but Katerina is working on it. She left the room just to go like help him get in. I think the thing is maybe he might use her account instead of his. So we might see Katerina's face, but hear Dr. Maldonado's voice. I think that's what it might happen. So let's see how that goes. Thanks again for your patience. We are going to get started really soon. I promise.
But we all know if Katerina's taking care of it, it's all pretty much sorted. She's always really great at having things well
Oh,
planned
hi,
out. So,
Katerina. Is it
oh,
Katerina or
there we
Dr.
go.
Maldonado? Who is it?
I'm Maldonado.
Oh, hey.
Welcome, Doctor.
Hi, but
Finally.
I'm in the phone. I don't know how it will be possible to go to my computer because I had my presentation in the computer.
Oh man.
I may I try with the link?
Yeah, and also, is it possible for you to maybe open your computer and put the phone next to it? Like just put your phone next to your computer as you're speaking. Is that possible?
Yeah, yeah, I think so. Yeah, yeah. I have now the computer very close from the phone very close from the computer. So what should I do now to to open the session from my computer?
Okay. So the slides, do you have slides or a presentation or
Yeah,
something on your computer?
I do have a slide. Yes.
Yeah, yeah. So just open that and just read from it or speak from it. We can hear you clearly. So whatever you say, we can hear it.
Okay. So without, so I present without slides.
Oh, yeah, you don't have to
Yeah,
share your slides on Clubhouse. We already have the link to your paper pinned at the top. So
Okay.
yes,
Okay.
yes,
Okay.
yes.
Sorry. I thought that was a classical presentation. Sorry, I
No,
was
no,
not
it's
aware.
okay.
yeah.
It's all right. Yeah, no, go
Okay.
ahead and present. But yeah, so we're gonna do a short introduction while you are getting settled. Yeah, so finally we're starting. Everybody, hello. Thank you for coming and thank you for your patience. This is actually Dr. Maldonado who is behind Katarina's profile. And so yeah, I'm just gonna give a short introduction of Dr. Maldonado and then we can get started. So, Dr. Rafael Maldonado, he got his degree in medicine, his MD degree from the Universidad de Cádiz, and also he got a PhD in pharmacology from same university. He also got another PhD in molecular pharmacochemistry from the University René Descartes in Paris. And then he also has a diploma to research direction from the same university. And so currently he is a full professor of pharmacology at the Universitat Pompeu Fabra. I think that's also in um it's in France also.
No,
And
in
he
Spain.
Oh, in Spain. I'm sorry. Yeah, it's in Spain. Yeah, that kind of sounded like Spanish to me as well. So he's carried out research for 11 years in France and the USA, and since 2000, the year 2000, he's a professor of pharmacology, like I mentioned, in Spain at the University Pompeu Fabra, where he founded the Laboratory of Neuropharmacology, which presently has around 30 people under his direction. His research is focused on the study of neurochemical slash anatomical basis of drug dependence, including opioids, cannabinoids, and nicotine. Moreover, he's studying affective disorders like depression and anxiety, and neuropathic pain and eating disorders. He has over 350 scientific articles in international journals, and he has been the principal investigator for 25 years of research grants funded by the main Spanish, European, and USA agencies. He's also a reviewer, a member of the editorial board of several scientific journals, and he has also collaborated with public authorities and private companies in the research policy and pharmaceutical development on drug abuse and pain. So he has a well consolidated experience as a group leader and investigator in neurobehavioral projects using animal models in an integrated approach. It is an honor to have you here, Dr. Maldonado. Thank you for coming. And so, usually, before we start the presentation, we just, sometimes want to have some questions about you personally, because that was your professional background. So, yeah, I want to know, Jamie, are you open to asking Dr. Maldonado about his background in science?
Absolutely, Dr. Maldonado. And we won't take too long, but thank you very much for being here on behalf of Science Society. And first of all, I'd like to just ask you, when did you first discover a fascination and affinity for science, and at what time in your life did you see this as being your future?
Well, that was long time ago. That was when I was starting my PhD. That was a project devoted to opioid addiction. At that moment, it was the heroin epidemic in Europe and in USA. and we found some specific components, some compounds. We were investigating benzodiazepine at that moment, and we found one particular benzodiazepine, flunitrazepam, that produced a dramatic effects on opioid dependence. And for me, that was the first passionating result that just inspired me just to go to do a science career, because I'm medical doctor. I have a medical degree, and at the beginning, you can imagine that my idea was to do the medical practice. And finally, I'm here doing science.
That's incredible. Thank you very much. And one last question, and then we'll get into it. Is what was the story behind you leading to this paper here today? What got you on this track specifically?
For this particular paper, the first point was the controversy about food addiction, because food addiction is really a very controversial issue, and we just proposed a challenge that was to set up a model in mice, in rodents, of food addiction. And once that we finally develop this model of food addiction in mice, we start this new project that I'm really also very, very passionate with this new project.
Thank you very much for sharing that. We always love hearing a bit of the background behind it. Now, thank you very much, Dr. Maldonado. Now, when you're ready, just relax, and we're ready for your talk. Thank you so much.
Okay, thank you
And
very much.
also, yeah, no, I just wanted to say, are you just let us know if you don't mind maybe questions as you're giving your talk, so you can have interruptions and then we ask you questions, or if you want to wait until the end and then we can ask you questions. So whichever one works for you.
No. I prefer the question in the middle.
Okay.
At the moment that there is any question, anything that you don't understand, please interrupt me and ask me the questions.
That's perfect. Okay, thank you. Well,
Thank
there you
you.
go. Go ahead, please.
Okay, so I just to very briefly present this new paper that appeared 2 months ago on Journal of Clinical Investigation, that is called microRNA signatures associated with vulnerability to food addiction in mice and humans. So first, to underline that this is a collaborative project. We are in the University Pompeu Fabra, but other groups in University of Girona, University of Barcelona also contribute to get this final result that has been now published. So what was the starting point? The starting point, as I said, was the controversy about food addiction, because the concept of food addiction was already defined in 1956 by Randolph. At that moment, let's see, almost 65 years ago, it was defined that some individuals, some particular persons with some particular food can develop a disorder that mimic drug addiction. That was in 1956. But today, we are in 2022, and the last manual of psychiatric disorders, the DSM-5, doesn't include yet food addiction. So you see, the controversy is still open. Food addiction, it is really a mental disorder. It is not, and this is a real controversy, So if we go to DSM-5, to the concept of substance use disorder, there are 3 hallmarks of addiction that are defined in the DSM-5. These 3 hallmarks are the persistence in seeking the drug, the motivation for drug consumption, that means the drug is really the center of the life of the person, and third, the compulsivity. That means the person is taking the drug in spite of the negative consequences of drug consumption. So this is in DSM-5. But as I say, food addiction is controversial. It's controversial, and there are food addiction scores to define this disorder in humans. And there is the Yale Food Addiction Scale that has been redefined recently using exactly the same criteria than for substance use disorder. That means persistence to seek the drug, motivation to seek the food, motivation for the food, and compulsivity to taking the food. And so we have in the paper that we publish now in Journal of Clinical Investigation, we use this model of food addiction in mice. So how is possible to develop food addiction in mice? For that, we use the operant behavioral models. We use the Skinner boxes. Now remember, in the Skinner box, the animal, the mouse, learn a behavior, in this case, in order to obtain a reward. So to develop food addiction in mice, we need first a particular kind of food. Which one? Chocolate flavored pellets. Mice love chocolate. They really love chocolate. And they become addicted to this particular flavor, to chocolate flavored pellets. So, but we need a really very long period of training. In our operant model, we train the animals during a period of 115 days, almost 4 months. You can imagine the length of the training in a mouse. A mouse, as you know, just live for a period of 2.5 years. So we are training the mice during 4 months in order to obtain this food addiction. And how do we evaluate persistence? Persistence to seek the drug again and again, we evaluate just by taking a 10-minute off period in the middle of the operant session. That means the operant session is on during 25 minutes, suddenly the light disappears, and during 10 minutes, the session is off. An animal that persists to seek for food is still making active responding in the Skinner box during this 10 minutes period. And here is the way to evaluate the persistence to seeking the food. How do we evaluate the motivation? The motivation we evaluate with a classical progressive ratio schedule of reinforcement. It consists that the first time that the animal press to obtain food, the animal must press only once. To obtain the second pellet, the animal must press 5 times. Third pellet, 12 times. Fourth pellet, 21 times. And this is increasing. And there are mice that press 2,000 times in the active level just to obtain one single pellet. This is a extremely high motivation to obtain the pellet. You can imagine a mouse pressing 2,000 times in a lever. That's happening in animal that has a extremely high motivation for the food. And the compulsivity, that is the resistance to punishment. We use a fixed ratio 5. That means the animal must press 5 times in the active lever in order to obtain one chocolate flavor pellet. So in this compulsivity session, when the animal presses time in the active lever, he receives a food shock. And when he presses the 5th time, he receives another food shock and then the pellet. So you can imagine the compulsivity. There is really a negative effect because the animal receives 2 food shocks before receiving the pellets. So using this protocol, we are able to classify animals in resilient or vulnerable to develop food addiction. And so normally in a normal population of mice, you can get animals that get addicted when approximately 22 23%. Is the normal rate of addictive behavior in the normal population of mice, of Swiss albino or black cis-G mice, that are the two backgrounds, genetic background that we usually use in these studies. So in a previous study that we published last year in Nature Communication, we identified by using DREAD techniques that one particular brain area, that is the medial prefrontal cortex, and one particular cell type, that are the glutamatergic projection from the medial prefrontal cortex, play a crucial role in the development of food addiction. So in this previous Nature Communication, we identify the brain area, the medial prefrontal cortex, as a key area for developing food addiction, for the control of this food addiction. And taking into account this result that we previously published in Nature Communication, now we ask what are the specific changes occurring in the medial prefrontal cortex that lead to this food addiction. And in this study, we have focused in one particular epigenetic component, that are the microRNAs. You know that the microRNAs are non-coding small RNAs that modulate the expression of the genes. How do the microRNA modulate the expression of the gene? When a microRNA is present, normally it's going to promote messenger RNA degradation, or just to suppress the translation of the translation of the RNA. So the presence of these microRNAs is going to produce an inhibition in the gene expression. And normally microRNA are polyvalent because they are going to play with a large number of genes. So an up or a down regulation of a particular microRNA is going to modify a lot of different genes. So using this protocol of long operant training in the Skinner boxes, in the case of the Juno Africa investigation, it was during 92 days, we categorize mice as resilient or vulnerable to develop food addiction. And we just took the extreme population. That means the extreme mice that develop all the characteristics of this food addiction, and the extreme resilient mice, those that are fully unaffected by this loss of control, those that really has a maximum control of their behavior. So we have these two extreme populations: mice that are resilient to develop food addiction, mice that are vulnerable to develop food addiction. And we took these two extremes and we analyzed in the medial prefrontal cortex the expression of the different microRNAs. And what we found? We found a lot of microRNAs that were down-regulated. Remember, microRNAs are going to inhibit protein expression, to inhibit gene expression. So the majority of microRNA were down-regulated. And there were some few microRNAs that were upregulated, but really in not very consistent way, because later when we did the validation of this finding by RT-PCR, the validation was possible with those microRNAs that were downregulated, but was much more difficult with those that were upregulated. So by informatic analysis, we underline, we from the list of microRNAs that were downregulated in the mice vulnerable to develop food addiction, we identify 3 particular microRNAs that could be relevant for food addiction, that were the Myrna 29C, the Myrna 137, and the Myrna 665. And
these were the results that we obtained in mice. So now, in collaboration with the group of Girona, with José Manuel Fernández Real, he did the same behavioral analysis in humans. That means he applied this Yale food addiction score in a large cohort of humans, and he select those humans that were resilient to develop food addiction, that really control very well the behavior through seeking food, and those that fulfill all the criteria of food addiction. And we did it in parallel. That means the clinician, they focus exactly in the same criteria that we focus in mice. That means persistent to response, persistent to seek the food, motivation for the food, and compulsivity for the food, seeking the food in spite of the negative consequences. So using exactly the same behavioral approach in animals and in humans, the clinicians just analyze the microRNAs. Of course, microRNAs were the plasmatic microRNAs. Remember that microRNAs are present in the different brain areas, of course, but there are also circulating microRNAs that are at the plasma level. So just by measuring these peripherally circulating microRNAs in humans, José Manuel Fernández Real, he found exactly the same, very similar, exactly the same, no, but very similar microRNAs that were downregulated in humans that were vulnerable to develop food addiction. And interestingly, I forgot to say that our experiments were done in male mice. The clinician found this downregulation of these microRNAs mainly in men, not in women. So there is also a gender, a sex equivalence in animals and in humans. And from the different microRNAs that were identified in humans, there was one that where the correlation was particularly dramatic. That was the microRNA 29C. So we have here results in animals and in humans that are really very similar. We did the same behavioral analysis, and in the medial prefrontal cortex of our mice, we found a downregulation of microRNAs that were also downregulated in plasma in humans that are vulnerable to develop food addiction. So until now, we just have an association of a behavior with a downregulation of some particular microRNAs. But now we must demonstrate that these microRNAs indeed participate in the behavior. How may we do that? By using mice, of course. By using mice, we identify, of course, the first microRNA of relevance that merits to be investigated was the microRNA 29C that was in perfect correlation in humans and in mice. So we use a classical research strategy that is the tag decoy. The tag decoy means that you administer this decoy that is going to target the microRNA 29C, and you are going to produce a down regulation by using adenovirus. By using an adenoviral approach, you just produce a down regulation of the MIRNA 29C in the area of interest. What was the area of interest? The prelimbic medial prefrontal cortex. The area that we identify as the most directly involved in the behavioral responses mediating this food addiction that was in our previous nature communication paper. So now we administer this tag decoy in the prelimbic medial prefrontal cortex, and we produce a downregulation of the MIRNA 29C, and we evaluate the behavior. Remember that a mouse vulnerable to develop food addiction is going to have a downregulation of the MIRNA 29C in the prefrontal cortex. So our hypothesis is that if we decrease the expression of MIRNA29C in the medial prefrontal cortex, we are going to generate a mouse that is vulnerable to develop food addiction. And indeed, our hypothesis was confirmed by using this tag decoy approach. Because as you can see in the paper, when you downregulate the MIRNA29C, you produce a dramatic increase in the persistent to response, a very important increase also in the motivation, but interestingly, compulsivity was not modified. So if we, in this case, animals were trained during a short period, because our hypothesis was that animals are vulnerable to develop food addiction. So we don't need the 4 months of training, just 1 month. And after 1 month of training, you can see that the normal animals, the animals that just received the control injection, only 15% of mice develop food addiction. However, if you produce a downregulation of MIRNA29C in the prefrontal cortex, half of the mice develop food addiction in a very short period, in only 1 month. So these results validate MIRNA29C as a key component of food addiction, in particular in terms of persistent to response. The second microRNA that we validated was the MIRNA665. And the result was also very nice here, because as you can see, persistent to response was not modified, motivation neither, but look, compulsivity. There was a significant increase in compulsivity in those animals where you made a downregulation of the MIRNA 665 in the medial prefrontal cortex. And if you look at the total number of animals that develop food addiction after 1 month of training, you can see that only 16% of hello? Yes?
Yeah, hello.
Okay. So then we took the third microRNA candidate, that was the 137. And here the result was negative. There was a tendency to increase persistence, to increase compulsivity, but finally was not significant. So this third microRNA that we suppose that could play a crucial role, finally we were unable to validate the role of this microRNA, because when we downregulate the microRNA in the medial prefrontal cortex, we don't modify the behavior. So you can see that by using this approach, that was a translational approach, using both rodent approaches and clinical studies in humans, we identify specific signatures of microRNAs, signature of microRNA in the medial prefrontal cortex in mice and at the plasmatic level in humans, that are both closely correlated in mice and in humans, and that may allow to identify the vulnerability to develop food addiction. So this microRNA, we suggest that can be used as possible biomarkers to define the vulnerability to develop food addiction. And we also identify the MIRNA29C as a crucial component in the crucial mechanism in the persistence and the motivation to seek the food. And in contrast, the MIRNA 665, this MIRNA plays a crucial role in the compulsivity developed during food addiction. So the different MIRNAs play a different component in this complex behavior that is called food addiction. And the identification of these specific signatures may help not only to identify possible biomarkers, but also in a future to identify possible new therapeutic approach. Because remember that now we have a lot of novel medicaments that are being developed in order to just modify the expression of the messenger RNA. So this approach, we think that in the future could probably be a possible approach to treat in the future food addiction. And I think that this is what I would like to explain.
Wow, thank you so much, Dr. Maldonado. That was wonderful. Oh my gosh. My mind is definitely blown learning about all these things that you have just shared. And I'm pretty sure, everyone in the room probably feels the same way. And so, I mean, you were saying we should stop you in the middle to ask questions, but you're so good at explaining that I personally didn't have a ton of questions that, that were more about understanding. And because I just also didn't want to interrupt you because you were going so well. But I think now we can get questions. If anyone is on stage has any questions, please flash your mics. And also, if you're in the audience and you have any questions, please come up on stage and ask your questions, or you can post them in the chat and we can ask them for you. Okay, so we have someone coming up. But Jamie, Gilbert, Dr. Shaw, do you have any questions?
Oh, I actually do. Sorry, Dr. Maldonado, I was finding the mute button there. Your talk was actually incredible, and like Sister Rahim just said there, you were very, very good at explaining a lot of things. One question I did have for you was that when you say how this became a how the eating disorder issue became a bigger problem, especially through COVID. Does this mean external factors like psychological ones actually can affect the actual behavior of the microRNA and everything? Because if you're giving biological reasons and yet you mention an external reason makes this worse, is there a connection?
Yeah, well, this epigenetic mechanism, remember that is just the link between environmental factors and genes. So this is environmental, psychological factors and genes. So our hypothesis is that this epigenetic component, these microRNAs, are really the link between the behavior, this, as you say, this psychological tendency to lose the control and the expression of the genes. It's a mechanism that could represent the link between genes and environmental factors.
Thank you very much, Doctor. I have more questions, but I want to free the stage up for the audience. So, Sister Rahim, who's next?
No, Gilbert, do you have any questions? Dr. Shaw? If not, oh, go ahead, Gilbert.
No, I'm enjoying listening in so far. No questions yet. I think Aslak has a question.
Okay, yeah, let's go ahead. Aslak, do you have any questions?
Yeah, thank you so much. It was really interesting. I actually only got part of I came a little late to this, but super interesting. So just my question is that, is there some, does this mean basically, no, how would you put this in is there a clinical kind of a protocol that could be of value coming from this research that you would see coming up?
For me, for us, the clinical value of that is the identification of a biomarker. If you know that there is a vulnerability in humans to develop a loss of eating control, we call food addiction, but everybody at least call loss of eating control, and you are unable to know who is going to lose or not eating control. And so, if imagine that just by taking a plasma sample, you can have a biomarker of a possible biomarker of the vulnerability to develop this loss of eating control. For us, this is the most the clinical value of this result. the possibility of find in a future, of course, a biomarker needs to be verified in large cohorts, and this is just a proposal. But this is for us is the clinical value of our result. And as I said, the second clinical value in a future could be the possibility to develop novel treatments. We have now the messenger RNA that everybody has used to be vaccinated against COVID-19. So we propose that probably in a future, just by increasing the microRNA expression, you could inhibit this loss of eating control. But this is in a future, of course. In the present, the biomarkers.
Okay, thank you.
Oh yeah, go ahead, Dr. Shah.
Yeah, thank you so much, Doctor. That was absolutely fascinating work. And my question from you, because we know that the role of the 29C in a different pathway, and specifically about the inflammatory cytokines. And I was just wondering, did you came up with any results that that can be related to the activated of the T cells alongside with your research or not?
Yeah, you are fully right. This is the study that we are doing now, and we expect to submit the paper rather soon. We look into 29C. One of the main target, as you explained, is all the inflammatory component, all the inflammatory pathways. So we think that, and this is also a general trend today, that this disorder, as well as other disorders, there is a inflammatory component that plays a particular role. And this is the study that we are now performing. We are trying to see if modifying the inflammatory component, if inhibiting the inflammatory component, we are going to be able or not to inhibit this loss of eating control. And we have some preliminary promising results in this aspect.
Perfect. Thank you so much. And also, did you find any relationship between the genders, different genders, like a male and female, because it's an animal model, and in relation with your hypothesis or not?
This is also the study that we are doing now. So now we have in the paper we have male and females, but in humans, but we only have male mice. So now we are doing exactly the same. Well, exactly the same. No, we are doing a similar study in female animals to see the sex differences in the development of these behavioral disorders. You're going to see once that we publish this result in females that there are really striking differences in males and females. So the sex difference is very clear. For this behavioral disorder also.
Wonderful. Thank you so much.
Just a comment to that I've seen some good research evidence that in social intervention or group interventions for using CBT and other psychological methods, there's also a clear difference in same-sex groups compared to mixed groups in group therapy sessions for obesity and overeating and undereating.
Yeah, of course, in the gender difference in humans is clear, is very well accepted. You just look for if you go through the eating disorders, you go to bulimia, anorexia, you see a gender difference that is really dramatic, and the behavior, the loss of eating control leading to obesity. There's also a very clear gender difference. So I suppose that the psychological interventions will, of course, also depend on the gender.
Thank you so much. Dr. Maldonado. I have a question for you from the chat from Sharon. It says, do you anticipate this research becoming more granular with capacities to mute the gene that predisposes one to food addiction? If not, what challenges do you foresee that could potentially come from targeted gene therapeutics?
Uh, as I say, for us today, we are not on the genes. We are on the epigenetic intervention. So the way, but of course, probably you're right. Probably just targeting some particular genes, you can also obtain the same. But our result don't identify a particular gene. Just identify, as was underlying the question before, some particular pathways, the inflammatory pathway, for instance, that seems to play an important role here. So for us, the message could be try to go a little bit upper than the gene. Go to the regulator of genes pathways. And the regulator of gene pathways are the epigenetic markers, are the microRNAs. So, and today it's easy to modify microRNA expression. So in the same and gene therapy has some. If you modify the gene, you know that you have, from a clinical point of view, you have more limitations than if you just modify the RNA expression of the microRNA. So I think that in a medium future, in a long future, who knows, no? But in a medium future, I see more close the modification in the messenger RNA or in the microRNA than the specific modification in the gene. Is my point of view.
Great. Thank you so much, Dr. Maldonado. I know it's like about a minute before the top of the hour. Do you have a hard stop at the top of the hour, or do you have maybe a couple more minutes for maybe a couple questions? I mean, it's up to you, whatever works for you. We can go with your timeline.
For me, it's fine.
Okay, maybe around 5 more minutes. So Jamie, if you have one more question, I also have one, but yeah, after you ask your one question, I can ask mine too. Yeah. Thank you so much.
Okay, great. Thank you very much. Hi again, Doctor. I was curious when I was reading your paper, when you were talking of mRNA 29C3P keeping the behavior and vulnerability going in 669-3P. One thing that stuck out to me with that is, are these two mRNAs sort of working together to create this this situation for people? Like one of them's kind of causing it, and one of them's keeping it going, in that kind of state. Or am I understanding that properly?
Well, this is a multifactorial disease where there are a lot of components that explain this complex alteration in behavior. And what we identify is that if you see in the paper, when you do the principal component analysis, the PCA analysis, you see that in the PCA1, that is persistent to response and motivation, is the MIRNA29C that is involved. Whereas in the PCA2, that is compulsivity, is the MIRNA 665. So I'm speculating now, but if I speculate, I will say there are people where the principal component 1 will be more important for the alteration in the behavior. And in these people, probably you have the down regulation on the 29C. Whereas in those where you have a predominant compulsivity, probably, and I insist I'm speculating, probably you have the down regulation of the 665. So I see these two microRNAs as a, let's say, independent mechanism to finally arrive to a complex disease that is loss of eating control, that is food addiction.
Fascinating. Thank you very much, Doctor.
Yeah, thank you so much. I think someone wanted to come up and ask a question. Um, If not, I'll just ask my. So, do you know what specific genes are regulated by the 29 and the 665? Like, how do they relate to food or um, behavioral changes, if that makes sense?
Genes that are controlled by these microRNAs are related to behavioral control and are also related to inflammatory, well, mainly the 29C, to inflammatory pathways. But there is no, and also there are previous studies in drug addiction that also consider the possible relevance of 665 in the development of drug addiction. So looking the previous literature and the genes candidate, you have, I will say, mainly two main pathways. One, the behavioral, the behavioral control in one side, and in the other side, the inflammatory component. And these are probably the two main physiological or physiopathological pathways that could be modified by genes that are targeted by the 665 and by the 29, C.
Thank you so much. That makes sense. So, behavioral and inflammatory. Thank you. Thank you. So we're gonna have one last question from Kuprik. Hi, Martin Kuprik, if you want to ask your question.
Yeah, I'm just wondering, and thank you for sharing all of this. This is very interesting research you've done, and you guys did, you people did, not just guys, I guess. And my question is this: how is the food industry interested in causing such addiction? Is it possible to stimulate the RNA to the point where you would induce large populace to perhaps seek such compulsive behavior? This is very abstract question. Hopefully you can maybe answer it to it. And the second question is, in your opinion, the general populace, is there an interest from the food industries to reduce such addictions? I hope you get my sentiment. Thank you.
Well, very very general questions, eh? But I think that the food industry is interested in selling food. That's clear. As much as you sell, as much as you win. That's clear, no? But I think that there are not. This is here we don't study the components of food because we went to the most easy way. That is the chocolate flavor pellet. Is the most easy. And we just went to the easy, no? To the easy. Just modify the flavor. Modify the flavor. So what is clear is that by modifying flavors, you are able to develop loss of eating control, to develop food addiction. And of course, there could be some interest in modifying the flavors in order to get this loss of eating control. But in our study, we cannot say anything about the different components of the food because we haven't studied that. And I think that the general population, of course, is interested in this addiction problem, in this food addiction problem, this loss of eating control problem, and the food industry is doing his job, and that is selling food products, selling as much as possible, and we must to take care about this kind of food where there is this particular flavor easy to get at it, no? I'm talking about the fast food, all this kind of food where you start with a caloric component that is not so different from a general food, but that you get this particular flavor that start you to losing eating control. And this is probably the first way just to go to a general loss of eating control and to go to an obesity problem.
Thank you, Dr. Maldonado, for that. I didn't even think of it that way, but that's definitely a really interesting way to look at it. And thank you, Kuprik, for that question and looking at it from kind of like a globally, outside perspective. So thanks for asking that. I think we actually have one last question. Jamie is going to ask just one question to wrap us up, and then, we can let you go and hopefully have Katerina come back into her account. But yeah, Jamie, go ahead.
Thank you very much, Doctor. My question was, when you were saying in your paper that the modulation of the endocannabinoid, sorry if my pronunciation is terrible here, and the dopaminergic signaling systems in this pathway confers vulnerability to food and cocaine addiction, it made me wonder, does this mean that the actual causes of both drug addiction and food addiction can actually be quite tightly tied together, so that the possible solutions for one might mean a possible solution for others, for the other?
You are fully right. I think that you are fully right. You are mentioning our previous Nature Communication paper, where we reveal that the endocannabinoid system was a neuropsychopharmacology paper before, no? Where we reveal that this regulation of the endocannabinoid system in the prefrontal cortex seem to play a crucial role in this food addiction. And in the Nature Communication paper, we reveal that the dopaminergic pathway to the D2 receptor seem to play a crucial role. So you jump immediately to drug addiction. And suddenly you found exactly the same mechanism. If you see my trends, well, an old paper, Trends in Neuroscience in 2006, where we discuss all our findings with regards to CB1 receptors and CB1 cannabinoid receptors and drug addiction. That it seems that the endocannabinoid system is a common neuronal pathway, is a common mechanism for drug addiction to whatever drug is. So now we find results that seem to tell us that the endocannabinoid system is also playing a crucial role in food addiction. And dopaminergic receptor, you can see the very old paper showing the crucial role of dopamine in drug addiction as a common neuronal pathway, common neural mechanism for drug addiction. So dopaminergic and endocannabinoid system, we postulate that both are indeed common neurobiological mechanism for both drug addiction and food addiction.
That is absolutely fascinating. Thank you so much for your time, Dr.
Thank you.
Yeah, thank you so much, Dr. Maldonado. So we are gonna wrap up now. Thank you, Dr. Maldonado, for your time. So we're gonna let Katerina know so she can come back. But before that, I just wanted to thank you for I know it was strenuous, Dr. Maldonado, going through all this. Technical difficulties, but thank you for coming in and spending your time with us and even going over a little bit to answer all our questions. We really appreciate you. And we hope you can come back. We hope you can come back and, kind of maybe this time, with your own account and we can work things out. So thank you.
With less technical difficulty. Yes, thank you very much.
Yeah,
And
no, definitely.
can I
um,
take this? Oh, can I just take the chance to also say to you, thanks very much for coming. And here at Science Society, we're wishing you all the funding and all the best you can get for this incredible work. And we're very, very grateful for you being here. Thank you, Cecilia.
Thank you very much for your attention.
yeah, thank you.
Bye.
Bye. Yeah, Katarina's gonna sign in soon. And so, while she's coming back into her account, we just want to thank everybody and maybe give a few announcements. So, this week we have another speaker coming in on July 11th. His name is Dr. Oh no, sorry, never mind. Tomorrow, 9:30 a.m. Dr. Spontak will be talking about self-disinfecting and anionic polymers. So this is going to be really interesting to come and see. On July 11th, we have Dr. Shen. We'll be talking about the continuous Bose-Einstein condensation. So it's almost like, a science society, we like to switch gears between different pillars of science, different disciplines in science. And so today we had something about mRNA, and then tomorrow we're going to have something about condensation and Einstein and all that stuff. So there's fun stuff coming up. We hope to see you back here. And again, we apologize for what happened at the start of the room, trying to get everything sorted. But hey, it's an app. We don't own it. There's only so much we can do. um, to make sure we're getting this information out to you, but it is an absolute pleasure to host these rooms for you, and we hope you know that we really love doing this. So yeah, thank you so much. Jamie, do you have any last words?
Just thank you everybody for coming, and please subscribe to Science Society if you haven't. And as Cecil Rahim also said, stay tuned for all the different disciplines. We are more like the mixed martial arts of science. So,
That's funny.
um, so everybody have a great time with your day, and thank you for joining us.
Yeah, absolutely. I'm gonna close the room now in 5, 4, 3,
Bye,
2,
everybody.
1. Bye.